All right, welcome everybody. Good afternoon. And this is the second day of ISFS 2023. We're gonna start today with our science session 3. We're excited to bring you this science session focused on biochemical and biophysical processes. I am Tigis Tamer. I'm a postdoctoral associate at MIT, and I'll be moderating the session with Dr. Ruchika Bajaj from UCSF. We will be hearing from 4 speakers today for 15 minutes and then followed by 5 minutes of questions. And Dr. Bajaj will be asking the questions for us. Our next speaker is Dr. Lisa Eschen-Wilson from Scripps Research Institute. Dr. Eschen-Wilson aims to characterize how biological motors communicate within the body using cryo-electron microscopy, among other techniques. Her goal is to use this knowledge to provide avenues for therapeutic interventions in various diseases such as cancer, heart disease, and neurodegeneration. Today, Dr. Eschen-Wilson will be presenting her talk titled Visualizing Dynamic Molecular Machines Using Cryo-Electron Microscopy. And with that, take it away, Dr. Eschen-Wilson.
Thank you so much for that lovely introduction. I'll just share my screen and get started. All right, amazing. So thank you again to all of the organizers and to Yale and the ISFS community. I'm so grateful for the mentorship and the community that we're building. And I'm very grateful to have the chance to share my work with all of you here today. So the title of my talk is Visualizing Some of Nature's Most Mysterious Molecular Machines Using Cryo-EM. And really the star of our show today will be YME1L. And so this is an inner mitochondrial membrane protein within the mitochondria, and here you're looking at it pictured on the inner region, so the inner membrane of the mitochondria. So this is really just to give you a quick glimpse into how amazing and versatile this protein really behaves, and this will be the focus of the talk today. So if I had to synthesize my passion and my thesis around structural biology, it would really be that form follows function. So structure provides a great deal of insight into an enzyme's role in the cell, and that has really been what I've seen over my entire career. So starting with my PhD at Berkeley, where I focused on microtubule regulation and how different enzymes can modulate the mitochondrial function and creating specific populations, like Dr. Krauss very beautifully illustrated in the talk before me. And through newfound collaborations here at Scripps, I've also seen how cryo-EM can be a very powerful tool to study viral entry and set up structure-based vaccine design platforms, which has inspired me with structure-based drug design for my future work. And that really lends itself to this, again, the core story today, that is the role of this ATP-dependent protease in mitochondrial quality control and health, called YME1L. That'll really be the focus of today. So mitochondria are double membrane organelles, and I really want to emphasize that because that means that maintaining quality control and health has to happen independently from what's in the cytosol. So it's isolated from the ubiquitin and proteasome traditional cytosolic quality control mechanisms in place. So what's happened is it's very beautifully evolved really an army of proteases to be able to maintain quality control and health within the mitochondria and its different subcompartments. And one that really stood out to me and drew me to the Lander lab here at Scripps was YME1L, because not only is it a protease, it's a hexamer of ATPase and protease domains, but it's also able to pull apart huge aggregates, site-specifically cleave. It has this great deal of structural plasticity and conformational plasticity. So as a structural biologist, I was very interested in this, but also how that alone is playing a role in maintaining quality control and health. That also very much drew me to this project. So YME1L within yeast has been well characterized at the stage of substrate translocation. So what this means is when a misfolded protein or protein that's targeted for degradation needs to be degraded, YME1L will pull it and intercalate along the amino acid backbone and pull it into its proteolytic chamber. And it couples ATP hydrolysis to be able to do this beautiful work. However, this is really only one stage of its full mechanochemical cycle. So we understand, as well as other labs, how it's translating these substrates, but how it engages, how it recognizes, how it's able to release substrates, how that changes under stress, is it tissue specific, how does that change as we age? These are all open questions within the field. And what excited me, given my experience studying highly flexible and dynamic proteins previously. So how do we get from sample to structure? First, we purify it out of the cell, and then we are able to design a construct that has all the domains we're interested in. So for this project, we're focused on the catalytic domain. It is a full-length membrane protein, but to start, we really wanted to look at just the catalytic domain and be able to see all of its possible conformations as a starting point. And so here I'm showing the construct design, and we actually have a pseudo transmembrane domain that's known as a coiled helix, and that helps hold everything in place because that transmembrane domain is really essential. But we can create this fabricated version to first start to identify or to start to see just the catalytic domain in action in vitro. So then we plunge freeze the sample. And so this is really where I spend a lot of my time day to day, is just creating grids and making grids. This process is extremely rapid. So we plunge freeze grids actually at 100 meters per second. So it's a very fast process. So here I'm showing you a grid that has my sample, I blot, and then it's rapidly going to reach the liquid ethane. And then I take that out and wash it in the liquid nitrogen surrounding that liquid ethane pot in the center. And so this really helps us create vitrified ice, almost like a glass-like ice, so that I can use the Arctica to image this process and image my protein of interest. And then I can start to collect on a high quality detector, a high quality camera, and get images of this protein in action. And so I want to give you a summary of all of these different conformational states that I was seeing at sample, which I found very impressive. And so I want to highlight that you're seeing almost like a stop sign, full hexamer, versus that stop sign getting completely split open. And then even here, you see it as well, where there's now this large opening, which was really unexpected because the protease domains have very tight interaction. So the energy required to pull that apart is substantial, and it alludes to this other state, this open state, as we started to call it. So I started to narrow down along this trajectory of possible states, where would the 3 most likely states be? And that really helped me narrow it down to pursue high resolution structures of each of these very dynamic motion-filled proteins. And so there was the open state. Here you're looking at top 2Ds and then a schematic where I'm just imagining the ATPase is getting split open and what I was seeing in the 2Ds. Then this capture state where it's starting to wrap around your substrate, followed by this closed state that's been well characterized in the field. And you're starting to see that intercalation and degradation of your target protein. And so using motion-based neural network models, it allowed us to connect the dots and be able to get structures of each of these states, which was really critical for putting our story together and seeing these proteases really split open, because there can be artifacts in the 2Ds, and it's really not until you get your structure that you can feel confident that what you're seeing is real. So we are very excited to have all of these different points and start to tell the full story, or start to propose a full story. So what I'm imagining is you have the protease domains and ATPases in this open state split open. And then once you have substrate come into close proximity, you can imagine YME1L starting to lock it into place. So you can imagine it's like a little Cheeto and now it's getting ready to chew it up. It's really excited. Then it's starting to position its prey into the center and decide or read it and say, okay, is this really, do I want to target it? Do I want to make a commitment to destruction? And then in some cases, yes, I do. I want to chew it up and I want to degrade it, versus site-specifically cleave it or chaperone it. So single-handedly, this protein can do all these different activities, and we believe that this is due to its conformational plasticity that cryo-EM is allowing us to see. So the main takeaway messages that I want you to have today, or take away today, is that now you can observe the full conformational and functional landscape of the yeast YME1 complex. And so we also have additional biochemical data. I'm not going to show it to you today, but very happy to discuss more in the Q&A. But it supports that these conformational rearrangements are required for cleavage activity. So when we mutate, for example, there's a critical glycine that is really important for the ATPases and the protease domains to make this motion as well as this motion. And when we mutate it, we see that cleavage activity move away or get abolished. And so what's really important there is that these rearrangements alone are contributing to activity, which is very exciting to see. And then finally, we're seeing this open state actually conserved across species. So there was a bacterial homolog solved previously using X-ray crystallography, but this symmetry that you see across the split stop sign, where you have these three ATPases and proteases on one side, and then the equivalent on the other side, the field thought maybe this is an artifact, or how do we know what this means? But then seeing it so nicely in yeast as well is providing evidence that this is not an artifact. This is a real possible conformational state that's critical for its activity, and potentially supports this endosymbiotic hypothesis that these mechanisms have been conserved and brought with the mitochondria from its past. And so thinking about future directions, really the next step is looking at this in humans when we want to think about drug development and its role in disease. As are these states observed in human YME1L? Can we learn more about substrate specificity when I talked about positioning it and deciding how it will read it? So is it sequence specific, or is it reading the fold? If it's misfolded, for example, is that a cue that it knows to be able to degrade it? And then finally, YME1 here pictured, I'm showing you that it can degrade so many different kinds of substrates, including substrates that are critical for mitochondrial fission and fusion dynamics. So these critical dynamics that we see are very important for health. And so how does it sense these dynamics, and ultimately, how does it affect mitochondrial communication? And so that really lends into my future lab vision, which is modulating mitochondrial crosstalk to meet high energy demands. So I get so excited about harnessing cryo-EM, cryo-ET, and cellular assays and biochemical assays as well to really focus on high energy tissues. Research shows that in these tissues, you have more clusters of mitochondria talking to each other, regulating whether we need to put out more energy and faster. And I'd love to catch that in action, both in healthy and in disease states. And to be able to ask, how are mitochondrial networks built and sustained? And how can I track potential modulators that are great targets for drug design in the future? And so on the last slide, I just want to go through my 3 key questions for the future that will be overarching for my lab. And the first will be, what mitochondrial structural features and behaviors in the eye are modulated to meet its high energy demands? What about the eye allows this to happen? And then what molecular players modulate the formation of what's called the mitochondrium? So in addition to these networks, you also have networks of networks, and then you have a total volume of mitochondria, which I think is really exciting to start to tease apart what molecular players are feeding into this huge network. And then finally, how can we build rational therapeutics that affect people who are suffering from mitochondrial disease that does affect nearly 2 million people? And I'll also be at the intersection of vision science that affects billions of peoples, of people around the world. So with that, I'd love to end and thank my amazing colleagues, my postdoc advisor, my graduate advisor, my other amazing advisor here at Scripps, who is Andrew Ward, Gabriel Lander, Evan Nogales, and then of course all the members of my lab. And so with that, I'm very happy to end here and take questions. Thank you.
Thank you, Dr. Lisa Ashen Wilson. It was such a nice talk to correlate your structure and function of this proteasome activity. So before I move to the questions from audience, I would also like to ask some of the questions,
Oh,
like
yes,
for example, hi Lisa. So so the first question I was asking, like, I missed it during your talk, that did you add ATP ligands in your sample, because it's a ATPase. So I was wondering, like, how did that affect your conformational analysis or so?
Great question. So for this, we wanted to add just ATP because we're looking at the wild type catalytic domain. And so in the past, you can add ATP homologs like that will slow down ATP hydrolysis or freeze hydrolysis so that you can capture you have less activity in place. But because we wanted to see the full conformational landscape, we took on this very ambitious question to look at post-hydrolysis states. Oh, that's my timer, sorry. So what was really interesting there, and why your question is so important, is we added only 1 millimolar of just ATP and saw that very almost immediately, because we freeze after just a few minutes, that all the two states that I show, the large open state and that capture state, they all have only ADP. So they've run through all their ATP, and now this is a post-hydrolysis state. And so trying to image that has been a challenge for the field because you have so much motion. And so usually you want to do exactly like you're saying, you want to have these ATP mimics that slow down hydrolysis so you can freeze things in place. But we wanted to be a little crazy and do the opposite and have it eat all its ATP, just have ADP, and try to capture the swinging motions of all the domains. So, but great question. So we just added ATP.
Yeah, so I do have more questions as well, but I will first go to the questions from the audience. I think people are liking your talk a lot. So first comment and question is from Guillaume Orteco. So he or she says that generally speaking, is the structure more conserved than sequence across evolutionary homologs? In general, how applicable are structure insights when looking across species?
Yes, so for yeast versus human, there's almost a 90% sequence conservation, but for bacteria versus the others, there's actually, they only have one protease that's called FTSH, fits is kind of what we call it in, like what I call it, but it does the work of multiple proteases that we see humans proteases do. So it is much more simpler, and there is a structure of that. And we can see that the catalytic domain is nicely conserved, but in terms of this motion and seeing that, that is what we were excited to see conserved. Because even though it has simpler roles, it's still using this conformational plasticity, which I think is this very nice evolutionary tool to do more with less. Because if you can imagine in the cell, there might be limited situations, like especially in disease or cancer where you have hypoxia, you have limited oxygen, maybe you have limited ATP. Can you still sense and engage with substrates? Can you still do some proteolytic activity? And maybe it's using these other states to do that. So very good question. And then, I'm sorry, what was the second question? There were two questions.
Yeah, sure. In general, how applicable are structural insight when looking across species? Because species may have just minor differences. So that's what the question is, like how structure helps that.
Yeah, exactly. So I think in the case of yeast versus human, no one has yet been able to see the transmembrane domain in action because it is so challenging to isolate the full length. So I do think there is a great deal of similarity between both species, but focusing on human is important for drug development that is applicable and physiologically relevant. So that's a great question. Why not just do yeast full length and try to answer all your questions there? But we really want to move into human because of what it can do for us functionally in terms of developing drugs. and testing mutants, and especially focusing on the eye, because you might imagine that YME1's activity varies based on mitochondrial populations or mitochondrial mitose subtypes. So they might have different machinery sort of protocols in place based on where the mitochondria is in the cell and then where the mitochondria is in the body. And that's also something I'd really like to look at, focusing on humans. But you're absolutely right that looking at yeast because of the close conservation is a great contingency plan, which I will also do in the future. Absolutely.
Yeah, so Liza, there are two more questions I would suggest, talk about. So Krishna says, great talk, Liza. What role, if any, do you imagine post-translational modification having on YME activity? Is this something you want to look at in the future?
I love that question so much. So it's not something we've touched here in the lab or that the lab is focused on, which makes me excited because I can do it in the future myself. So I think that, yes, like to answer your question, yes. And that was actually my first PhD project, was looking at post-translational modifications on microtubules and how that changed structure and function, and also how that helped create subpopulations. So I want to do the exact same thing now, but with mitochondria. How are post-translational modifications on these proteases, starting with YME1, affecting its activity overall. And what's really crazy about YME1 actually is that it's hypothesized to interact with other proteases. So just imagine like Transformers version, they all come together, or maybe the Power Rangers, you know how they come together and they make one huge machine. Okay, so I imagine this for proteases, and YME1 is part of that. And I'd love to test how PTMs affect YME1 and how that affects its assembly overall with cryo-EM, but also now with cryo-ET, because that proposed assembly is big enough to see by cryo-ET potentially. So that's what I would love to look at. Great question. Yeah.
Hi, Lizard's last question along the similar lines, that is SOMO domain of YME simulated? Might YME be regulated by redox state? And another question from the last point, you said that it's also available for cryo-ET now. So is that like, can you watch that in the cell itself now? Considering the molecular weight, like, can you tag it and then check it out? Like, how big is that? Like, can you comment on that?
Yes, I think this person and I need to talk more.
No, so the cryo-ET question was mine, but the simulated was, yeah. So,
yeah, very good question. So for the first question, it is affected by that PTM, and for cryo-ET, what's really important is that we want to see how YME1L is organized in the ultrastructure of the mitochondria. So how PTMs affect potentially how it's decorating the cristae, or how it's affecting positioning of fusion proteins. So very specifically, YME1L regulates a protein called OPA1, and that structure was just solved by Adam Frost in Nature in August. And they do a really beautiful job showing how, or proposing how it's being organized in the cristae. But what's sort of the next step is what puts them there. And if YME1 is critical for making the active forms of OPA1, I just imagine it's right next door to these proteins at the cristae, helping regulate or move it to where it needs to be. And so PTMs, like the one you mentioned, can very much be involved. I'd love to test that. I'm nowhere near that yet, but I would love to test that. So great, both really great questions. We should talk more.
Thank you, Liza. I think we are getting late for another speaker, but yeah,
yes.
I am a membrane protein biologist by training and ABC transporter. So I think it will be good to talk to you.
I would love
sometime.
To
About the ATPase and then cryo-ET stuff. Yeah.
Oh, that's wonderful. Yes. Thank you so much.
Yeah, thank you. I will move to the next speaker. Yeah.
All right. What a lively discussion. Thank you, everyone, for, participating through that. Our third speaker today is Dr. Eric Villalon-Landeros from Johns Hopkins University School of Medicine. Dr. Villalon-Landeros studies proteasomes in the peripheral nervous system, or the PNS, and his aim is to investigate mechanisms that are relevant for development and function, the PNS in health and disease. His talk title today is Nociceptive Activity of Peripheral Sensory Neurons and Modulated by neuronal membrane proteasomes. And with that, Dr. Landeros, take it away.
Yeah, thank you so much for that kind introduction. And I first would like to start by thanking the ISFS organizers for putting this event together and giving us this opportunity to present our work. And so, as TJ said, I am a postdoc here in the laboratory of Seth Margolis at the Johns Hopkins School of Medicine. And classically, I have been trained as a cellular molecular neurobiologist. And my interests in terms of research have been to understand how peripheral nerves develop and function. So for those of you that might not be necessarily too familiar with the nervous system, it is divided into two general parts: the CNS, that includes the brain and the spinal cord, and then the PNS, the peripheral nervous system, is everything outside of that. So all the nerves extending away from the spinal cord and the brain. And so throughout my PhD training and my postdoc training, I have been interested in understanding how these peripheral nerves function to allow us to move and interact with our environment. And so the story that I'm going to tell you about today is a fairly recent discovery that I have made in the lab here, and is basically the basis of what my future lab is going to focus on, and what we will try to study in the next several years. And so in the laboratory, we study proteasomes. And proteasomes are these large macromolecular proteins that are in charge of degrading proteins to maintain protein homeostasis. These proteasomes are made up of 4 heptameric rings, the alpha, beta, and alpha rings, that are organized in such a manner that allow it to make this barrel-like shape structure with a pore in the middle. And these proteasomes have 3 catalytically active subunits: beta 1, beta 2, and beta 5. And so this is the 20S proteasome. Now, this 20S proteasome can associate with regulatory cap proteins, like to form the 26S proteasome to mediate ubiquitin-dependent protein degradation. And so with an interest in understanding how these nerves function, one thing that we know is that these proteasomes are found in every cell in our bodies, in every cell, and also within all compartments in the cell. And yet we do not fully understand how this works, how these proteasomes work. As a matter of fact, most of what we know about how proteasomes work has come by the use of protein or proteasome inhibitors, such as those used for treating cancers in human patients. And one thing about these proteasome inhibitors is that they have one main side effect, and that is development of peripheral neuropathy. And this is the cause for patients discontinuing their treatment. And so this highlights the fact that we do not yet fully understand how proteasomes work in our peripheral nervous system, at least. And so my question when I joined the Margolis lab was to understand what is the nature of proteasomes in the biology of the peripheral nervous system. And initially, when I joined the lab, the Margolis lab was initially only focusing on studying proteasomes in the brain. And so when I joined the lab, I wanted to first see where these proteasomes are localized or located within the peripheral nervous system. And so I took a mouse as my model, and using 2 very specific antibodies, I took some tissue and labeled these, used those antibodies to see where our proteasomes are. And so using immunoelectron microscopy, and that's what I did. I used gold or antibodies that were conjugated to gold particles, and then use our primary antibodies on spinal cord tissue. And so here's what we found. So each one of these dark dots is points to the location of a proteasome. And interestingly, we found what we were expecting. We have these gold particles that localize in the cytoplasm of what we think are neuronal structures here. But then we found these other gold particles that localize to these dark lanes, which are what we think membranes of neurons in the spinal cord. Now, here we have some quantifications used to show the relatively high abundance of those gold particles localizing to the membrane. And in here, these violent plots just show us the distribution of those gold particles as we look away from the membrane. We can see that there's quite a high percentage of those gold particles on the membrane. And so this became very interesting. I also first initially looked at it in the neuropil and spinal cord, but then also looked at it at this localization in the dorsal root ganglion. So this is where the sensory cell bodies are localized, and found similar results. We see gold particles, or these dark dots, localizing to the membranes. We see the same thing in the axon, as you can see here. There are these gold particles localizing along the axonal membrane, and then we have some controls just to compare and contrast. But so this data is consistent with the idea of there being a proteasome that localizes on the membrane in our peripheral neurons. Now, this became very exciting to me for several different reasons. And one was because several years ago, back in 2017, the Margolis lab published this paper where, using similar approaches, they found in the central nervous system a proteasome that localizes to the membranes of cortical neurons. And it localizes there in a manner that allows for degradation of intracellular peptides and release of those peptides as extracellular signaling peptides that can bind on neighboring or nearby neurons and modulate their activity. And so from now on, I will be referring to this membrane proteasome as the NMP. Now, this the idea of having an NMP present in the peripheral nervous system, specifically in sensory neurons, was very exciting, because this could mean that this is a communication system that functions to allow neurons to communicate with one another, which has been a question for many, many years in the field as to why and how these neurons communicate with one another. And this would not be possible to have been discovered by even decades and decades of research using our regular proteasome inhibitors. And so this became very interesting to me. And now, then my question shifted to what is the role of the NMP in the peripheral nervous system? Now, to begin to investigate this first, then I wanted to see if I could detect these NMPs on the most distal parts of the sensory neurons, and that is in the skin. So I took the skin from the paw of mice, and using a non-permeabilizing labeling condition, I used my antibodies to label for proteasomes. And so here, what I'm showing you here is a staining of using our beta-5 subunit proteasome antibody, and we can detect this red puncta on skin sections, which we think are proteasomes. Now, these puncta localize and overlap nicely with nerves sustained here by PGP 9.5. And so to really show that these were proteasomes that are localizing on nerves, then I did a denervation experiment where I cut the nerve on the paw, or that innervates the paw of these mice, and allowed these nerves to degenerate away. And when I did that, I repeated the labeling experiment, and I found no puncta that also correlate with all nerves being gone. And so this indicates that, yes, these puncta are probably and most likely proteasomes that are localizing on nerve endings in the skin, or that innervate the skin. So then I wanted to investigate how this functions. And so to begin to do that, first I wanted to use a very specific inhibitor of the membrane proteasome, and that is biotin epoximycin. So biotin epoximycin, just by putting a biotin molecule on epoximycin, it makes it membrane impermeable. So it only binds to whatever is exposed to extracellular space. And so I then injected another set of animals with this biotin epoximycin, and under normal conditions, normal innervated conditions, I can see the incorporation of biotin over time in the paw of these mice. And now here I did another Western blot to look at the beta-5 subunit of the proteasome, and that is the subunit that the biotin epoximycin binds to. And I can see detection of the normal beta-5 subunit here in the darker line, but then I see this shifted band corresponding to what we think are proteasomes that have biotin epoximycin bound to them. So we can see that biotin epoximycin in the skin targets proteasomes. Now, to really show that those proteasomes, again, are proteasomes likely sitting on nerves, I performed that denervation experiment. And when I did that, I treated the animals again after the denervation, and I see no detection of biotin, and also no detection of this shifted band, indicating that when we remove the nerve, we also remove any membrane proteasomes, or these membrane proteasomes are actually in the nerve. And then I wanted to then use that to study in vivo in the system, how or was it, what is this NMP doing? So I took another cohort of animals and injected them into the right paw with biotin epoximizing and did some behavioral experiments. First, I tested for any changes in sensitivity to heat, and I found that treating them with biotin epoximizing basically has no effect. But then I tested for their sensitivity to touch or mechanical stimulation and found that this increase in 50% withdrawal threshold over time. Basically, the bigger this measurement is, the lower their sensitivity is. And so this indicated that injecting biotin epoximizing into these mice reduced their sensitivity to touch or mechanical stimulation. And I have several more experiments and data pointing to the idea that this is also involved in changing their sensitivity to pain sensitivity in these mice. And so at this point, it became really interesting and intriguing, and also unclear as to why treating these animals with biotin epoximizing would affect the function of a subpopulation of neurons, given that all neurons and all cells have proteasomes, right? So it became really unclear why we would only get a specific functional effect on these nerves or in these mice. And so the next thing that I wanted to see is to investigate whether the NMP is localizing to a subpopulation of neurons. And so we can do this. I did this by basically dividing all our sensory neurons into three main groups, and that is the neurofilament heavy group, so as labeled here in the green. And this group of neurons are large neurons that are generally in charge of sensing mechanical stimulation and proprioception. And then I did my antibody feeding or unpermeabilized labeling for proteasome using another antibody against the alpha-2 subunit, and that is here in red. And we can see that some neurons have this membrane proteasome, as you can see here in the picture on the left with the red puncta. And then some of this same group of neurons don't have the membrane proteasome, which was very interesting. And we see the same thing in other two groups, the CGRP positive group, which are C-type nociceptors, smaller neurons that are generally in charge of sensing pain due to heat. And then also in the IB4 positive group, again, these are C-type nociceptors that are non-peptidergic and generally in charge of sensing pain due to mechanical stimulation. And what I found here is that the IB4 positive group had the highest percentage of neurons that were positive for the NMP. And that was very interesting because it correlated with our behavioral results in that inhibiting the membrane proteasome affects mechanical sensitivity. And so I wanted to investigate this further using our primary cultures here. And so I used calcium imaging to do further studies. And so here's my stimulation paradigm. I first stimulated the neurons 3 times to activate them, and then incubated them with vehicle or biotin epoximizing, stimulated 3 times again, and then one last time with 15 millimolar KCl, and then one high KCl stimulus at the end. And so what I found is that incubating or treating these cells with biotin epoximizing reduces their response to stimulation, as you can see here in the pink curve. And then what I did is separated the responses into different size neurons and found that these effects are mostly localized or localized to smaller, small to medium-sized neurons, consistent with having a phenotype of pain and mechanosensation. And so then one question that came up was whether or not if this was a system that neurons used to communicate with one another, whether the proximity of them from one another was needed in order for the system to work. So I investigated this by basically taking our normal cultures that were seated at normal density and then diluting them to separate these neurons as far apart from one another to see if their proximity was needed, and then repeated this experiment here. What I found is that in our normal culture or normal density culture indicated here by the 250, just like we did before, incubating with biotin epoximizing led to a decrease in response to stimulation, as we can see here. But interestingly, just diluting the cultures alone reduced the response to stimulation, similar to inhibiting the membrane proteasome. And then inhibiting the membrane proteasome in the diluted cultures, as indicated here by the 50, had no further effect. And this suggested that this system is functioning in a way that allows for these neurons to communicate with one another, and it's functioning in a non-cell autonomous manner. So using what I've been able to discover, what I've been able to find, and what we know about the membrane proteasome from the CNS, I have put this model together where we have different types of sensory neurons indicated here by the different color that have membrane proteasome. Now, this membrane proteasome is found in the cell body and all along the axons, but just for simplicity, I'm only showing it here on the cell bodies. And basically, this membrane proteasomes release peptides, and then these peptides bind on neighboring neurons, and these peptides function to allow for normal calcium dynamics during neuronal stimulation, leading to normal neuronal activity, as indicated here by the black arrow. And so what happens when we block the membrane proteasome, then we don't get those peptides. And then calcium dynamics are dysregulated during stimulation, and that leads to reduced activity. And so that is the model that we have put together so far with my data. And so what my lab will focus on in the future are three initial questions. There are three big questions. The first one is to understand what are the peptides, what are these signals that this membrane proteasome is sending out from the neurons, and what their targets are, where are they binding on the neuron. That is not clear at all. And so that's one of the questions that I want to focus on in the beginning, to begin to understand how this system works. Then my next question is going to be, what leads to this cell-specific expression and localization of the membrane proteasome? How come some neurons have this membrane proteasome and some others don't? And in those neurons, in a cell-specific manner, is there a difference in the function of this membrane proteasome? Does it function differently in different types of neurons? And then my other question is to focus and try to understand the involvement of this membrane proteasome and its signaling peptides in the context of pain. Is it upregulating pain? Is it causing more pain in already pain conditions? Does it make it worse? And then again, in pain conditions, under pain conditions, does that affect the function of the membrane proteasome, its recycling, its localization to the membrane? And then can we use what we know about the membrane proteasome to see its effects on the pain already? If we block it, does it reduce pain or does it increase pain on pain conditions? And I can do that in using mouse models. And so that's going to be the if you want to call it, 5-year goal of my lab. The initial few questions, but then the long-term goal of my lab is to try to understand and how the system works, and leverage our understanding of the membrane proteasome to try to use this to come up with potential therapies or treatment approaches for pain conditions in humans. And so with that, I would like to thank the Margolis lab, especially Seth Margolis, who has been very generous and instrumental in me getting this research program put together and up and running. And so I would like to thank also all the members of the Margolis lab and Dr. Michael Katerina for also supporting me and providing me with advice here at Hopkins. And with that, I would like to take questions.
Thank you, Dr. Eric William Landros, for your such a nice mechanistic talk. And I am happy to take questions from the audience.
While the audience questions are rolling in, I actually wanted to ask, how does this relate to, or has there been evidence of these proteasome functioning at a higher rate, lower rate, and like in the context of addiction? So where patients or even mice models are like have a higher tolerance for pain medication.
Yeah, thank you for that question. And that is, like I said, like one of the long-term goals of mine, right? We don't understand. I don't know how it might function under addiction yet. The way we understand how it works is under normal conditions. Everything that we have done so far, is study this membrane proteasome on the basal conditions just to begin to understand how it's working. And now the next steps in my research are going to be to incorporate this into like other conditions like pain, and understand how if it functions similar, if it's functioning at a higher level. The idea that we have and how this works is that under normal conditions, the membrane proteasome is releasing signals normally, right? And then when there is inputs into the neurons and sensory neurons from the periphery or from pain conditions, then the function of it is going to change, and either is going to start sending out more peptides or less peptides to finely tune the response of sensory neurons to more inputs. And so, yes, that is a very important and critical question that I don't have the answer for you.
Okay, thank you for answering that question. There are two more questions related to your A2. What are you currently thinking is the mechanistic basis for NMP cell specific expression?
Yeah, that's also, thank you for that question. And so what I think might lead to cell specific expression is, so the way we know how this membrane proteasomes gets to the membrane is actually by associating with other auxiliary proteins. And so the mechanism by which it gets to the membrane only in some neurons lies on what these proteasomes are associating with in those specific neurons. So to understand how and with the mechanism how they get there, first we need to understand what they associate with, and are those auxiliary proteins cell specific. And so that is one of the questions that I have, and I don't have the answer for that. Like I said, this is a fairly new discovery, and we're just barely starting to understand how it works.
Thank you, Eric, for answering that question. I have another question for you. Maria appreciates that your work is so fantastic. And do you project your research to impact the development of bionic arms for naturalistic sensation? A main challenge is the Wallerian degeneration after an amputation or nerve injury. How do you believe this can help?
Right, and so I believe that if this system is really functioning to fine-tune the sensitivity or the function of sensory neurons in cases of injury, for example, with Wallerian degeneration, where the neurons, some of them die back. We know that when that happens, specifically under like an accident, for example, nerve injury, where some of the neurons die, the rest of the neurons, the rest of the axons become more like hypersensitive. And we think that this system might be functioning to mediate that communication between the neurons to make them more sensitive to stimuli or to inputs, or even under basal conditions. So if we can understand how this system is working, then maybe under conditions of an injury, we can either target the receptors, the targets of the peptides, and inhibit the system that way, or modulate the system in that way to decrease the neuronal sensitivity, or we can synthesize the peptides that are relevant for this system to work, and maybe inject those peptides into the system to, again, regulate it and try to manipulate the sensitivity of the neurons. But we still don't know how it's working under those conditions. You're muted.
Dr. Bajaj, you're muted, sorry. All right. Thanks so much for that discussion, everyone. Thank you, Dr. Landeros, for the wonderful talk. Our last speaker of the session is Dr. Stephanie Vankovich from University of California, San Francisco School of Medicine. Dr. Vankovich studies how drug candidates change the entropy of targets. Her aim is to make drug candidates that bind stronger to their target, and thus improving selection of specific drug candidates, as well as having a better chance of killing targets, especially in virology, such as SARS-CoV-2. Dr. Vankovich will be presenting today a talk titled Making Sense of the Chaos, Conformational Entropy, and Ligand Binding. Thank you so much. Take it away.
Thank you so much for having me, and thank you for the organizers for putting this together. It's been really wonderful being a part of this community, and I'm really excited to continue building community with all of my co-fellows. So my name is Stephanie Vankovich. I am a computational and experimental structural biologist at UCSF, and I study this concept of conformational entropy, which is something that has been really starting to blossom in the past 10 years as something that really contributes to the energetics and functions of proteins. And my work really aims to see how we can bring this into a structural biology field to take advantage of understanding structure, but understanding dynamics and understanding the energetics of proteins to be able to improve our ability to understand protein function, to be able to design new drugs, be able to design new proteins. And so my work has really taken quite a journey. So I really started in a very clinical setting, looking at patient response to different therapies, both in terms of their clinical outcomes, as well as DNA or RNA sequencing. And it's really moved into this basic biology field where now I can not only just understand a different mutation and sort of its relation to being response to treatment, but what actually are on an atomic scale, what is driving that? And so throughout my talk, while I'm very focused on sort of basic concepts of biology, this background that I have of working with patients and working with the translation is something that really drives me, and I hope to continue moving that forward in my own lab. So protein dynamics are essential for function. Proteins are not static, even though the majority of structures that we have are static. And not only are these protein dynamics important for the protein be able to reach different states, to be able to bind a ligand or bind another protein, or to move something across a membrane, they're also important energetically. So even when a protein is in a single state, even when it's in its native fold, it still can move a lot. And the movement actually contributes to the energetics of the protein being able to do its functions. And this is related through entropy. So the Gibbs free energy, which defines all protein functions, is made up, which is the delta G here, is made up of the enthalpic portion, the delta H, as well as the entropic portion, delta S. And we're really great as structural biologists at estimating and getting some hypotheses about what is going on in this delta H. You can imagine that if you have in a ligand binding sense, you have the ligand come in and you have the protein coming around, and you can look at the specific interactions. And we can make some hypotheses. Why does one ligand bind and why does another? But that completely ignores this delta S. And based off of this equation that we know underlies, the energetics that's driving these functions, we have to be able to investigate this delta H as well as this delta S in order to understand protein functions. And so where does protein dynamics actually connect to this delta H? This delta S? Well, it comes through conformational entropy in proteins. So as I said, even when a protein is in its native fold, it can still move around. And the majority of this movement is actually due to the fluctuations of side chains. So side chains can access many different rotameric states, and the fluctuations between the different rotameric states not only can exist within a stable fold, but it actually helps stabilize the fold. But it also provides the protein with this reservoir of energy that it can then use potentially to help transport something, help bind something, help do any of its functions. And recently, many different protein functions have been shown to be impacted by conformational entropy. And here I'm just showing a list, but this list is definitely not complete in terms of the different functions of proteins that are impacted by conformational entropy. And we also have information that this is linked to binding affinity. And so today I'm really going to tell you a story about the role that conformational entropy plays in binding affinity. And so I'm finally going to let you watch this entire MD simulation, which comes from D.E. Shaw. It's a Src kinase, and it's inhibitor dasatinib. And now dasatinib has bound the protein, but notice that the protein doesn't stop moving. And so this difference in the protein dynamics between when it's bound and when it's not bound is this difference in conformational entropy. And so if we're able to measure some of this in a structural sense from structures, we can get at what are the contributions of entropy in many of these protein functions, such as binding affinity. And we know that binding affinity and protein conformational entropy are related based off of this great study out of Josh Wan's lab, where they correlated protein conformational entropy measured by NMR on the x-axis and protein conformational entropy measured by ITC on the y-axis. And each dot here is a different protein ligand complex with different colors representing different proteins. And there's this nice linear correlation. But what NMR doesn't tell us is, how does the protein actually get this conformational entropy? I said it mostly comes from residues moving between different states, but NMR is just telling us that a single residue or a protein is moving more, but it doesn't tell us exactly how it's moving more. And so if we break this down into a single toy residue, you can imagine that you have this single serine side chain, and it can have conformational entropy by just sitting in a single rotameric state and it kind of like wiggling and jiggling around. But you could also imagine some other perturbation of the protein in which it now accesses multiple different rotameric states. And by accessing the multiple different rotameric states, it can increase its conformational entropy. And so if we're able to extract these quantitative metrics of conformational entropy from structures that are actually telling us, exactly how side chains are moving, how it's interacting with the enthalpic interactions of a ligand binding or binding another protein, we can really get at the mechanisms of what conformational entropy is doing, and importantly, be able to apply that prospectively to ligand design, to protein design, to understanding functions. And so what we really need to be able to connect this quantitative metrics of energetics and the structural mechanisms is we need to be able to model structures as ensembles. So structures not being static, they need to be modeled in their multiple conformational states. And this will allow us to get out this quantitative metrics in these structural mechanisms. And so my work has really turned to how can we extract conformational ensembles from X-ray and cryo-EM structures. So X-ray and cryo-EM structures are not structures of a single protein. They're actually the structure of thousands to billions of copies of the protein. And this is because in a single crystal or in a single grid in cryo-EM, you are taking those pictures and we are getting information from the many copies of the picture. And if a single protein in its native fold still can have residues that are fluctuating around and moving between different rotameric states, well, this data about conformational entropy should exist in X-ray crystallography and cryo-EM structures. But the issue is that this information is pushed into this really, really messy data structure of electron density. And this is mixed up with experimental noise, it's mixed up with solvent noise, it's mixed up with information about ligands. And so to be able to extract out what the conformational ensemble is of proteins from this data is very, very difficult manually. And so myself, along with a really great team, have been developing this tool called QFIT, which leans on some machine learning based algorithms to be able to model in a lot more of this noisy data instead of having to do it by eye. And so briefly, I'm just going to walk you through what we do is we break down the protein for every single residue and we sample backbone. And then we get all of the different potential conformations of a backbone and using some optimization algorithms match this to the electron density. And we do the same for side chains, sampling a bunch of different side chains, and then using similar algorithms to be able to match up what the experimental data is telling us compared to our model. And finally, we stitch the protein all back together, looking at clashes, and we're able to then label it. So we can see that there are multiple different conformations that a single protein is taking on. And so what QFIT does, and this is specifically focused on very high resolution X-ray structures, is it's able to find these hidden alternative conformations. So it's able to find the fact that in the experimental density, there are multiple positions of these side chains, and it's able to automatically model them in. And this really reduces the burden from having to make these structural ensembles. But importantly, over a large data set, QFIT and modeling structural ensembles actually explains the underlying experimental density better. So here I'm showing R-free, which is a metric of looking at a model compared to the experimental density. And anything below that red line means that QFIT is improving our fit of our model to our data. And as any, regardless of whether you work with structural biology, right, our goal as biologists is to try to design experiments that we can, get data, but then create models that are able to best explain this data. And what this graph is really showing us is that by modeling proteins as structural ensembles, we are explaining our experimental data better. And so that we need to continue pushing on this to explain the experimental data even better so that we can really hear and understand what our proteins are trying to communicate to us. And importantly, we can also do this type of modeling in cryo-EM. And I'm really excited how we can map this with some of the larger structural changes that we can now observe in cryo-EM on top of just a single side chain moving around. And so we have this multi-conformer model where we now can, model in side chains in multiple different positions. But importantly, this is what allows us to also get this quantification of conformational entropy. So imagine you have the same side chain, the purple or the green, and we are able to model it into multiple conformations, and we can look at the distance between how much the side chain is actually moving in the purple or the green to get this quantification of conformational entropy. And so now we have our information about our energetics and our information about our structure. And I think this will help us explain the mechanisms of a lot of protein functions a lot more, getting back to explaining this delta H and this delta S of energy. So the question I really wanted to ask now that we have our tools, what is the contributions of protein conformational entropy and ligand binding? And so what we did is we modeled over 1,500 different X-ray structures with a ligand bound or ligand not bound. And we asked on a residue by residue basis, what is the difference between conformational entropy? And what we saw is that across the protein, some residues increase their conformational entropy and some proteins decrease their conformational entropy upon ligand binding. But if we focus in on just the binding site, we observed that the conformational entropy is decreased in the binding site upon binding. And this would make sense, right? A ligand comes in, the protein then needs to hydrogen bond, or you have steric clashes, and the protein can't move a lot. And so we now have this model where, okay, so, the residues are able to move around a lot without a ligand, and then you have a ligand come in, and we know it restricts around the ligand. But what happens elsewhere in the protein? You can either have the entire protein become rigid, or maybe there's some sort of compensation where some part of the protein is increasing. And this is exactly what we saw. We saw that the greater the decrease in binding site entropy, the more it lost flexibility around the ligand, the more the protein tended to gain entropy elsewhere, distally in the protein. And so this leads us to this model where, the residues are fluctuating around and a ligand comes in and the protein compensates for entropy. And this makes sense, right? The protein does not want to lose entropy, regardless of any function it's doing. This would reduce its ability to do any function. And this is exactly what it's showing us. And it's likely that, this is clearly evolved, but it's something that we can take advantage of with medicinal chemistry. So we can understand sort of the native function, but also potentially improve drug binding. And so this is, very important to think about in drug binding due to the energetics. But the question is so in an individual protein, how are ligands then changing the conformational entropy? And so we turn to this protein of CDK2, where we had 13 different inhibitors found in different structures. And so in the rows are the inhibitors, the columns are different residues, again, colored red, more flexible, blue, less flexible, or increasing, decreasing conformational entropy. And there are some repeating patterns. But importantly, in the ligand aspect, you see there's two outliers. And this is because these outliers are type 1 kinase inhibitors compared to the rest, which were type 2. And so this indicated we can use this type of modeling not only to look at the quantification, but we can also look at how this is changing binding. And even if we look at the same protein in two different ligands, they have the similar energetics, but they change in certain residues where they're increasing their conformational entropy. And so moving forward, and this is supported by an independent NIH grant, we're now looking to see how ligands bound in the same site impact conformational entropy by looking at about 900 different structures of the same protein bound to different ligands, and asking how are chemical properties impacting differences in conformational entropy. And so the other thing, going back to my previous work, is I'm very interested in how mutations impact conformational entropy, and not only looking at, what is statistically different between patients, but how are these mutations driving these differences? And so just want to conclude with conformational entropy as a spatially reciprocal relationship, and different ligands to the same protein can have unique impacts. But to be able to get this, we really need these structural ensembles, such as QFIT or others, to improve our models of experimental density and extract those quantitative estimates of entropy. And so conformational entropy can fill in these many gaps into many of these protein functions, but we need not only we need the structural mechanisms to understand these functions. And so my future lab is really going to be working on tools and methods to model these structural ensembles. Asked how conformational entropy impacts binding and affinity, both looking at a genetic or a chemical landscape. And then asking how it impacts protein functions by looking at it, how it impacts signaling pathways, prospective ligand design, and prospective protein design. And with that, I'd like to thank my lab and the funding and take any questions.
Thank you, Stephanie, for such a nice talk and describing like how static structure can be really, really useful to determine the conformational landscape. Yeah, so we do have one question for you, and it's about how does conformational entropy scale with protein size? And also, does it increase or decrease when complexing with other proteins?
Yeah, so on the first side, we do have some data that larger proteins basically spread out their conformational entropy. So the amount of it doesn't change, but it will really, I think the spatial patterns become a lot more complex. And then the second question about protein-protein interactions, there hasn't been any studies about that data, but I would imagine based off of, there's a lot of studies of nucleic acid and protein and small molecules and proteins, and they all indicate that there is this spatial relationship where around the binding site it decreases and elsewhere it tends to increase. And so I would imagine that protein-protein interactions impact the same way, but that's something we'd really like to study in the future.
Thank you, Stephanie, for answering that question. So I was just on as a membrane protein biologist, I was wondering, like, have you tried membrane proteins on this methodology? I mean, they are pretty flexible, and I mean, they could probably show you more landscape than the one you have already started with.
Yeah, I have not, but yeah, I think that membrane proteins, they tend to have a lot of flexibility, especially right in like that membrane region. And I do think that has to do with sort of, they have this reservoir of entropy. So it would be really fascinating to look at that as well.
Yeah, we do have some like whole catalytic cycle of one of the transporter we have worked on. So if you want to talk about, like, I mean, we can talk about that with Jamie as well. Yeah. I think Ifan Cheng, I mean, is your neighbor, so probably they might have better ones. Yeah. Okay, so yeah, thank you everyone for joining us for the Biochemistry and Biophysical Processes session of IESFS Symposium, and well done to all speakers for sharing your exciting work with us today and also answering all these questions. It had been a pleasure to facilitate this session 3, and we are now going to take a quick break until 3 p.m. and we'll follow with the Fellows Science session 4 on communication, cells and beyond, and stay tuned. Thank you, everyone.